Thank you, Aline, and thank you, Hassan, for this nice introduction and having warmed up the audience also. So thanks. Well, it's a pleasure to be here today, and thanks to the organizing committee for inviting me, and that's a lot of things that we have already discussed. I suppose that the majority of the audience are people from development, but not sure. So here I'm representing the research part, so the very upstream part, before making any kind of API. That's important to know because at the API stage, all the properties are already fixed in the molecule. So there is no possibility to change it. At the very early stage, so we are in an iterative process. We are making molecules could be 100, 200, 300, until 1,000 molecules, until we are arriving to combine all properties, biological properties, the NPK properties, physical-chemical properties, in a way that we have the best API possible to be well absorbed and having the best properties for development. So not being the class 4. Anyway, so I explained what that's the different vision. So starting with the title, this is probably the title vision from the CMC partners, so from CMC Canadian Impact Research. I have another title which is from research, how to catch up the information from CMC to help us to integrate some technologies in order to control the absorption, because the main challenge, as you see, is solution, solubility, and absorption. So that is a part which is very complex, and we'll see in the process what are the different assays that we are using. Well, for those who are interested to know what the Kagaku Teki means, it's because I worked for a long time with the Japanese, more than 20 years. So I integrated that notion to my consulting company in a way that it's quite visible in Japan also. So there are two ways to write it, depending on the kanji, and it means or scientific or chemical. So when you are using the romanji, it's meaning both. Thank you. Well, the major goal of my presentation is to give the real experience. So at bench, with the hands-on of the medicinal chemist. So as a designer, producer of novel molecules, so it means that starting from a target, the pointer is working. Starting from the target, hit finding strategy, lead optimization, identification, and then optimization on one way, but redesign, rescaffold, resynthesis, whatever, and to, as a user of biological and DMPK in vitro and vivo data, to make interpretation of that, transfer those data into molecules, so changing the structure in a way that we are able to optimize. So those are probably the two missions of the medicinal chemist. Just to remind, so the research phase is something very progressive. We are going step by step. So it takes between 1, 2, or 3 years to get an API. This is a drug candidate. It could be a bit shorter, but it could be more realistically a bit longer, between 2 and 4 years. The second point is for each steps of this progressive process. So we have a number of checklists of parameters to reach and try to respect. So I will also speak about the causes of the major biased interpretation between biological data and DMPK in general. What is the integrated medicinal chemistry process? What we understand by integrated, and also what are the different critical potential issues to solve, and how the experience of development can be applied in a way, in a simple way, to research. So just to remind you, you don't have to read everything. There is two major phases. The first one is the very early stage, target ID, hit finding, hit ID until the lead identification. Then from that, we start again to our early optimization with many in vitro assays. Lead optimization taking account of in vitro, but also mainly in vivo assays, including PK/PD and so on, until we arrive to the drug candidate. When the drug candidate is selected, so it means that it corresponds to a checklist at least of between 15 or 20 parameters that we combine together to have the best candidate. So I would say classically, when you look at that, the early CMC, so the solid state, is generally evaluated very late. This is more the classical way. The classical way is that I introduce that at the early stage of development, but late stage of research. And for me, it's quite too late. It's quite too late because I lived in my own career a lot of different problems that could be solved before that. But it's not always in the mind of everyone. So that there is a part of the education to do, and we'll say, well, the education becomes with the failures. Right? The more problems you have, the more you learn. Well, so simply to say that in the discovery process, when you start with the very early in vitro, and until the in vivo PK/PD or chronic model really know, we are using more and more sophisticated assays. So it means that in the meantime, of course, we need to make resynthesis, one or two different batches, and batches are not always the same. Where are the differences? First of all, next, in practices, what we are doing stepwise. First of all, we are evaluating the activity, the selectivity, and then we start to evaluate the ADME property, the MPK properties in vitro. But we start with evaluating the solubility at least 2 different pH, 2 which is mimicking the stomach, between human and rodents, for example, and 7 the neutral pH. This is the first point, and this is very important because it's crucial for evaluating how we can solubilize the compounds from DMSO liquor, model liquor, to dilution, to solutions that we apply in vitro, and vehicles we have to use for the in vivo. So later on, now we are optimizing what checklist, the permeability, transepithelial models, metabolic clearance, and so on and so on. So each time we evaluate it, and the product that we are making are evaluated at the different stage. If not good, of course, we start again to modify and evaluate the change in the properties. The quantities for doing that are evaluating by in solution. That's not a problem. So in general, at the very early stage, products are coming from libraries, commercial libraries that are purity of over 80%. But in the other phases, we are increasing normally the quality, the purity of the samples, and we are going to solid samples. When this is in solution, there's one point I address a bit later about how to evaluate the real concentration in solutions, but mainly we are using samples, solid samples, between 10 mg to 1, 2, 5 gram, or even more for early SMC or chronic administration. So it means that between the batch 1, which was used here, early, and those batch, there is mainly differences that are not always evaluated. That's for sure. I can tell you by experience. what are the general discrepancies that we observed? There are two types. First of all, discrepancy between this data obtained with solutions and data obtained by solid sample that we put in solution, or something like a suspension or micro-suspension, or perhaps something which is a solution that we don't always very well evaluate. So the solubility is not really systematically evaluated. It's become more and more frequent, but not in all our projects. So something to establish in a very formal manner. Crystallinity and solid form is very often not verified. So batches and reproducibility in batches, normally perhaps visualization, look at our crystals, see whether they are really the same, but nothing else in general. Standard dissolution protocol, they are very often different between the biological studies and the different teams that are using, and the DMPK teams. So they are not using the same vehicles. So of course we cannot expect that the solubility could be different. These solubilizing vehicles could be also very heterogeneous. Some people are using MC or PEG or other different systems or mixtures, and others are using DMS or plus ethanol, or other systems. So, well, the impact of that is the solubility is different. So, and the one major point is the real drug concentration in the starting solution, and then after eventually different dissolution or dilution. So that is something which is very frequent in vitro, and it is something which is normally that can be evaluated easily by LCMS. But it is something that there are a lot of resistance by the researcher to do that, because they say, well, perhaps the LCMS to evaluate all solutions is a bit tricky, or it's overload work and so on. But in reality, there are many discrepancies between results, because the real concentration of the solution is different from one batch to another, or from one experiment to another. So when we calculate the IC50, they are not related exactly to the real concentration, but only to the calculated theoretical concentration. It could be wrong. So, yeah. Well, next point is just to remind you, not repeat what Hassan said. This was established well that this is exactly the same graph, so we use exactly the same publication. On the other way, well, when we are at this stage, this is the idea. That's what we have to focus on, right? Otherwise, if you are at this level 3, class 3, well, we know that low probability can be optimized in a way that working on polar surface in a way that we increase the permeability, the coefficient A to B, at least A to B, and not being B to a flux pump substrate. If you are at this stage, well, low solubility, we can work on that, making salts or reducing the lipophilicity, but it's already if it's highly permeable, it's a very good thing. Something we have to absolutely, totally avoid is the last one. So when it's at this stage and you are class 4 for the API, well, that's something for us that is not the goal. So we have to look at this, 1, 2, or 3. But since it is an iterative process, we know where we are at one stage, and we know what we have to optimize. It's always a combination of different properties. Well, just to remind that what is exactly what Hassan said. So it's something well established before. biology. In biology, so solubility being very critical. So what I said is the real concentration of solutions is very important. And you know well that starting from DMSO at 100 micromolar, so it means that you are making dilution 10 times, 100 times, 1,000 times, and that we suppose that the final solution is something that our concentration is 100 or 1,000 times less. But that's not always true. The physiologists, at least the vehicle for IV administration are very often different, but that's normal. I would say it's not always compatible because we have to use vehicles that are compatible with the route of administration, of course. Use of grass solvents, so we generally recommend it as safe vehicle, is something very important because that should be always compatible with the pharmacological models. Anyway, so here what I recommend is at that stage to make a vehicle screen. When we have something which is not very soluble, start to make a vehicle screen before making any kind of in vivo PK or PD. Otherwise, you know that you have to repeat that again. So, anyway, this one. So, in the chemistry experience, a long time ago, it's more than 30 years ago, 30 years, I have asked to my chemist, you are making your sample, and please don't look only at the pictures, but make a picture from microscope and see whether we see if the crystals are bricks or long or amorphous, or the solid is amorphous or whatever, but at least to know it. Because it's important to know what are the results related to that batch and what could be the solid state on the next batch. So create an historical reference in a way that we can understand if there is a problem later, where it's coming from. If you don't do that, you have no historical data, you don't know. You don't know where the discrepancies are coming from, so you have to start again from scratch. So, verify the forms, that's one way. On DMSO, verify the solubility after dilution and to quantify the real concentration, so by LCMS. When I'm speaking with different colleagues of pharmacology, DMPK, biology, and chemists about that, they say, wow, that's complicated. Well, no, everyone in the labs has a LCMS at the end of the bench. It's a routine work. That's not something very complicated. It's totally reserved for big pharma. No, that's something which can be done by anyone. So, necessary possibility, verify the solubility with classical, if not with classical vehicles, make a vehicle screen. Possibly make macronization, but that's something, we are speaking about macronization, we say, okay, like that. You need a kilo for making macronization. No. A few milligrams, 10 milligrams, that's enough. You are in very, very simple, system with different beads to break the crystals and to simply reduce the size of particles. It's quite enough to put that in solution. That can be done by a chemist at the bench. It's not a huge galenic technology. No, it's very simple. So, speaking about that, the chemist will say, it's not chemistry anymore, it's galenic. No, forget it. No. So, we have to change the education of verify the plasma exposure in that case, because crystals, forms, have a direct impact, and vehicles direct impact with exposure. What we need is the best conditions to have the best exposure in PK, corresponding to the conditions we will use for the PK-PD relation. For Buckleberry, what I found is out there very often a difference between the understanding of the different terms we are using between research and development. I'm not speaking about what, perhaps we are using the same words, but the significance is different. So there's something to catch up in research. For we are making a lot of many compounds to optimize. So it's an iterative process. As I said, 100, 200 more, quite more. So we are evaluating many properties. It's an iterative process, and there is no regulatory constraints. So we do that in a way that we are taking account of activity, selectivity, absorption, permeability, metabolism, and so on. We combine everything. As you know, in development, you are working with one compound. This is the API you have in hands. You are not able to change it. You can change perhaps the form, but well, this is the structure. This is the only one. So that's different. So it's a fixed process. You have always checklists to evaluate this, and it's highly regulated because it's a regulatory studies. So it's fixed. So of course, but we did a solid form. The aspect can be evaluated. Crystallinity is at particle size. This is a recommendation, but I say, well, that's something that is not always felt very formal. The solid formulation, probably never. So in research, never. In development, that's nearly 100% of the goals. So this is the other view. And finally, the kinetic solutions are sometimes used in research. I recommend to use, at least to evaluate the kinetic solubility, but it's made in a semi-empirical way as compared to development, where it's very well established to do that. You have very formal protocols. So, R&D connection. We know that the training between researchers and developers are a bit different. Even they have a common past, but at least the goals are different. They are in separated organization, sure, except perhaps in small biotechs or small companies. The departments are not on the same sites. The locations are totally different, but they can be even in different countries. The expertise is different. So in research, mainly medicine chemists, biologists, pharmacologists, early DMPK. While in development, you have more organic chemists making the process optimization, scalability evaluation, development DMPKs, galenic pharmacists, and toxicologists. So the training is different, and the people are not speaking very often together. So sometimes it happens. That's what I had in the past when I started working in medicinal chemistry in a private company, but that's not the rule everywhere. How to that research can benefit from development expertise. That's why we are here today. So that's very important because we can save time, we can save resources, and we can save expenses. And this seems to be obvious. It's not only a slide, but we are facing problems. Then we see that we have to repeat studies, that other people will tell about this. We have to repeat costly studies. We have to repeat that the solid state analysis, the PK/PD, and if you are coming to chronic administration, it's even worse. So it can cost not only a few 10,000, few thousand, but it's 100,000 euros. So this way we have to be very careful. Well, just a reminder, drug candidate nomination is when we are at the end of the process of the optimization. So small quantity synthesis, physical, biochemical, pharmacological profile in general, early PK, metabolism, very important to control the clearance, and safety profile. While this is more the research phase, and this is really the work of mainly the CROs. In development, we have other kind of requirements. Large-scale synthesis, certified batch, which is not made here. Solid-state analysis, which is not very often made here. Validated analytical methods, not done in research. And solid stability, not done in research. So the requirements are quite different. So there is something here, it's more the CDMO world. And well, that's different. Research, CROs, that are making services for research activities. CDMO that are totally focused on development. Some of these are, well, they are saying that they are doing both. CRDMO, I don't know. So after that, perhaps we have CRDMO and then something else. But well, you have to be conscious that nobody can do everything. So we are better to address the questions to professional and specialized persons. Wow, there is a gap. There is a gap between those. Drug candidate nomination, that's fine. So we arrive with investors to a point, we have the drug candidate. So we can enter into regulatory studies. Wow, but there is a problem. The problem is that, first of all, we need the scale-up batch. The scale-up batch. So how much we need? 10 grams, 20 grams, 100 grams, depending on what kind of study we have to evaluate. But well, scalability is not something you do that from 10 mix to 100 grams. No, we are using different steps to make it slowly, scalability from perhaps 1 gram to 10 gram. So whether there are more impurities, reduce solvent quantity, or optimize the conditions. Then from 10 grams to 50 grams. And then at the same time, the chemists are simply creating the historical data on the different batches. Crystallability, solid state, solubility, the different pathways, but also impurities profile, which is very important for the next step. Before if you have no batch, nothing can happen. No DMPK, no safety study, nothing. Next step, when the batch is available, now you start the solid state, the salt, or the solid state assessment. Before that, nothing has happened. So you simply generate different polymorphs, stability, whatever, and then you come to the end at, we have the batch. Already a scale-up batch which is available, and then we have selected the form. Then, when you have both, you can start to say, I select both, and with that batch I can make dynamic studies, safety studies, DMPK studies, and everything. But before that point, principle 1, and here, that can happen between 6 months to 1 year. Because you are facing, for example, multiple polymorphs. Polymorphs which are not stable at different temperature. Polymorph 1 is stable at room temperature. When you keep that, you heating that at 30 or 40 degrees, this is the polymorph 2 which is the most stable. So it depends on the crystallization process. When you are going into the cake with the development scale-up batch, and you are drying the cake, you are heating it. So perhaps at that stage you change from one polymer to another polymer. This becomes a nightmare. So that's why it's taking some time. No? So this is real promise at the bench. So you can write, we have Gantt chart plan. We say we can do that point. Yeah, but I'm facing the reality. that is something different. Yeah. So nothing happened before you have the batch and you have selected the form. Solid form, but we know that's something obvious now, that poor solubility is a real challenge. It's not something we have to repeat. So what are the different parameters that are driving the solubility? We know at the research data that TPSA, so polar surface, is one element. Log P, log D. Also pKa, of course. Also, that's something we can evaluate with predictions, in silico predictions, but also calculated and or measured. That's something we can do at the research phase. Solid form, it's not what could be that the compound is amorphous. It can be crystalline, something which is very often missed. Is the batch 2 a solvate? Is that an hydrate? Is that a solvate hydrate? Or is that a mixture? So even sometimes we are asked, what is the molecular weight of the sample? And they gave the molecular weight of a neutral compound, but the batch is not the neutral one. It's called solvate hydrate or bisolvate hydrate salt. So the molecular weight is when we are weighting the compound. So it's not the same weight. And the IC50, the concentration is not the good one. So the IC50 value is wrong. So that's something to keep in mind. So the analytical data of each batch, which just an airplane is fine. So we have to take care of this. We can even have more aggregates and different forms. Well, the last point, I don't think that perhaps you just mentioned that the packing energy in the crystal, the higher is the energy. It's making bricks. So you have that crystal. I crystallize it. Wonderful. So I got very nice crystals, but solubility, that's a problem. Yeah. So making nice crystals is not a good goal. No, it's nice. It's nice for science, but it's not good for development. So we know that depending on different crystallization conditions, we can work on the way we add anti-solvent and so on, and reduce the size of crystal or change the crystal unit form. The three major processes of discrepancy between biology and the MPK data are coming exactly from the same criteria or the same parameters. Kinetic solubility is different because they are working on different batches and or different forms and particle size. And the dissolution protocols and vehicles are different. People are not speaking together. Or sometimes, in that laboratory, they are using classical mixture of that. In the DMPK services, they are using another. And they will say, but it's a bit difficult to change the routine work and so on, so have to ask them. So, anyway, so the impact of that the data are perhaps sometimes comparable, but sometimes not comparable. So the scenario is that I connect again on that point. There are real concentrations in solutions. There's something to really establish at the early stage, because I had a few examples of really wrong data. So in the real practice, first of all, you have to know that 20 years ago, many people were crystallizing that product. It doesn't exist anymore. Crystallizing? What does it mean? No. So we are using purification cartridge in a parallel system, and also evaporating the solvent, and then you have something which is solid. Then a solid could be amorphous, could be crystals, whatever. So, and of course, the solubility will be different. One example I really had in mind, because that was dramatic for the project, is the hair inhibition to the potassium channel, which is responsible for QT prolongation. For a drug was established at IC50 at 5 macro. I believe we have the recommendations, something with IC50 over 10 micro. That's what we have. We were at 5 micros in good trend for optimizing, but in reality, when we evaluate the effect, because we use the guinea pig model to evaluate the QT prolongation, we had a very used QT prolongation. I thought, what's happening? So when we verify the real concentration of the drug in the condition of the hair inhibition by patch, in fact, the concentration, the real concentration was quite lower. So we got 50%, not at 5 macro, but at 0.5 macro. So it means that we are quite over the threshold. So that's good, huh? Immediately. So that's a direct acting point. Now let's simply envisage that I think easy and high throughput technology of CMC can be applicable at the research phase, and that is why I really try to promote something which is coming from CMC in a way that that could be applicable at the early stage. Let's go quick. Wow, absence of solid form. Wow, repetitive mistakes. It's always the same. So absence of historical data, that's a problem. Lack of data on the form, non-reproducibility, and discrepancies. When you see that, wow, that's really nice. So consequences that use time and money. Wow, we don't have to rediscover the wheel. No, everything is existing already. So it's simply to catch up from development. So everything on form stability, solubility, parameter in the form, so on. Everything is already available. It's simply to make a formal checklist or a formal process in a way that this is applied in research. As you know that even if you are using very often solutions, these are coming from solid samples. So solid samples are dissolved in different vehicles, DMS and so on. And what we recommend is simply to evaluate at least solubility, examination of the solid form, visualization, and make a simple solid form evaluation as soon as we are at the lead stage until we scale up batches to verify that we are really always in the same balance. Yeah. Conclusions. Solid state analysis in research and discovery is largely underestimated. It's not a routine work. It's sometimes exceptionally some people are doing that, but not a routine-based. We have to convince the people that do that. It's a benefit. It's really a benefit for research. The other point is the experience of development can be very useful. I think you are totally convinced. And medicinal experience could be really crossed with solid form analysis at the early stage if the technologies or the equipment are very easy to use on a routine base. And then I think that's something we have to think together, bring people together, think together in a way that we can have different stage of simple evaluation of solid state and so on, until very complex, which is probably the CMC requirement, but not apply the CMC requirement as such, but in a simpler way that would be used on a routine base for every kind of problem we are doing. You need to know that chemists can do per year 100 compounds, 200 compounds per year, one people. So you need something which should be very easy and quick. Not quick and dirty, but quick working. Thank you for your attention.
Thank you, Pascal. So we take the time perhaps for a very short question, if there is a question. Yeah, it's okay. Question? No specific question on this talk. So we, yeah. So we will start this second session. Relative to oral formulation current challenges. So please. So we will start. So we will start the second session with Julie AUFFRAY. So thank you for being here today. So Julie is a postdoc at Bordeaux University in the team of Pierre Choroloff on interdisciplinary projects, including galenic developments and especially applied to biologics. So that is the subject of her talk today. So related to development of biologics in tablet form, a new approach for local delivery. So thank you.
Thank you, Aline. And hello, everyone. And first, I would like to thank Skyepharma for this kind of invitation. So, as Aline said, I'm Julie AUFFRAY. I'm a postdoc researcher at the University of Bordeaux in two different labs. The first one, which is specialized in galenical development for solid dosage forms, and the other one in biology, but more specifically in microbiology. And so today I'm pleased to share with you the work we are doing in these labs, but more specifically what our efforts to develop a tablet for biologics that you will see represent a very promising approach for their local delivery. So just before we begin, I would like to quickly remind everyone what biologics are. So biologics are distinct from conventional and chemical drugs, how they represent any compounds or any molecules which derive from living organism. So biologics can include, for example, any compounds or any molecule used in cellular and gene therapies, any immune modulators, any vaccines, or any monoclonal antibodies. And as you can see in this graph, the share of biologics that you here in yellow has grown significantly over the past decade, and nearly reaching 30% of all the approved drugs in 2023. But for this presentation, I choose to mainly focus on monoclonal antibodies, which represent the largest part of biologics, and that demonstrated maybe the most impressive increase in the pharmaceutical market over the past decade. And just to give you an idea, in 2024, MABs accounting for 9 out of the 15 biologics that has been approved by the FDA. In 2023, they accounted for 12 out of the 17 biologics approved by the FDA. So just to remind you that antibodies are a normal constituent of our immune systems. They are very large protein with a wide-shaped structure that are produced by immune cells, but more specifically by plasma cells. And they play a very important role in our immune system by neutralizing pathogen and also by coordinating the immune response during an infection or during a disease. And there's many reasons that can explain why MABs are used in therapeutics. First of all, they present a very good safety profile. This good safety profile can firstly be explained by the fact that a MAB possesses a very high specific affinity for a unique target, which limits the risk of side effects. And also thanks to the recent advancements in genetic engineering, which progressively made possible to develop fully humanized MAB, so limiting the risk of immunogenicity reactions, and which makes them very safe when they are used in clinic in humans. The other reason, and maybe the main reason why MABs are used in therapeutics, is because MABs possess diverse natural functions. For example, MABs possess a neutralizing activity, which can be useful when, for example, you want to neutralize a pathogen during an infection, or to neutralize a high immune modulator, that could be involved in a disease. Also, thanks to their ability to opsonize and to activate a mechanism which is called ADCC, MABs can be used in order to coordinate an efficient high immune response with the other high immune cells. And finally, MABs can also be used in order to modulate the high immune response, for example, by stimulating the activity of other high immune cells. And this is thanks to these diverse natural functions that today MABs are used in a wide range of pharmaceutical applications, such as in oncology, in immune-related disease, or also to treat or prevent some infectious disease. So now let's discuss the formulation for these MABs. So MABs are proteins, which means that they are very sensitive and susceptible to degradation in the gastrointestinal tract. But also MABs are very large protein and hydrophilic molecule, which means that they have a very limited intestinal absorption. And this is maybe for these two main reasons why the feasibility of developing a MABs formulation for oral administration has been really quickly rolled out. And so today, MABs formulation are exclusively available as a solution or freeze-dried powder for a systemic administration. And while this type of administration present many advantages, such as providing rapid systemic bioavailability of the molecule, for example, it also presents many drawbacks. As an example, it implies to use aseptic pharmaceutical manufacturing process to be developed. It also induces the maintenance of the cold chain during the storage, especially for solutions. Concerning patients, it represents a not very well accepted type of administration, which most of the time requires a medical personnel to do the administration. And finally, and maybe the main drawbacks, is associated to pharmacokinetics aspects, because as we said earlier, MABs are very large protein and are very hydrophilic molecule, which means that when they are diffused into the systemic circulation, they present a very limited distribution to the diseased organ and to the target antigen, which means that sometimes it's necessary to administrate a large amount of the molecule to observe the biological activity. So this is very problematic, and even more when it comes targeting superficial conditions where there's no necessity to have the MAB in the systemic circulation, and where the MAB have to cross multiple barriers to reach the antigen. There are in fact many different situations where the possibility to directly deliver MABs at the mucosal site could be very judicious. For example, it could be the case during an infectious disease. I'm sure that most of you know that during an infectious disease, the first port of entry of the pathogen are mucosal sites. And though in this clear case, and for example, during a viral infection, the possibility to directly deliver the MABs at the mucosa could be very judicious to obtain an early control of the infection by directly neutralizing the pathogen at the mucosa and so limiting its penetration into host cells. Also, the possibility to directly deliver MABs at the mucosa could be judicious to control some other non-invasive infections, such as, for example, vaginal infections, such as vaginose or vulvovaginal candidiasis. As another example, we can cite inflammatory bowel diseases, and for example, ulcerative colitis or Crohn's disease, which both are characterized by chronic inflammation of the gastrointestinal mucosa. Today, these diseases are treated with MABs that target a cytokine, the TNF-alpha, which play a key role in the activation of the inflammation. So these MABs are used in order to reduce the inflammation and to modulate the immune response. And in this case, it has been demonstrated that the biological activity of these MABs were more associated to a local activity rather than a systemic one. And also we know that the systemic exposure with these MABs can induce some adverse effects. So in this other case, the possibility to directly deliver MABs at the mucosa could be judicious both to limit the risk of adverse effects, but also to increase the biological activity of these MABs. So I hope that with these two examples, you see that there is a clear necessity to develop alternative dosage form for mucosal delivery of MABs. And that's why in our labs, we try to explore the feasibility of formulating MABs in a high innovative form, which is the mucoadhesive tablet. The mucoadhesive tablet, in addition to offering the possibility to directly deliver the MABs at different mucosal sites, presents also different advantages. First of all, it's a dry dosage forms, which can offer a high stability for biopharmaceutics. Using mucoadhesive tablet could also be judicious to simplify the supply chain without the need to use aseptic process, without the need to maintain the cold chain during the storage of the MABs. It could also be judicious in order to improve the patient compliance. And finally, it represents an interesting form when you want to control or modulate the release of the drug. So to obtain a tablet containing a MAB inside, it's a two-step process, where the first step consisted to try the MAB, because as you know, when the MAB are produced, they are in solution. But as you can see, this first step was not very limiting during our study, because currently 25% of the commercially available MABs formulation are already developed in freeze-dried powder. However, the second step was more limiting. So here you can see the general pharmaceutical tableting process. And as you can see, it implies applying very strong force on the powder in order to obtain a cohesion. So it's easy to understand that the tableting process is a very high stressful process that is supposed to affect the integrity of biomolecules such as antibodies. So that's for this reason that the main objectives of this project was to identify if they exist, because at the beginning of the project we didn't know if it was possible. So to identify the key formulation and process parameters that could ensure the preservation of the biological activity of MABs when formulated into tablet form. So this project, we were able to carry out this project in our labs thanks to the equipment we have, which include a freeze dryer and a compaction simulator that both are fully instrumented, which means that we were able to control any process parameter during the two steps of this development. Well, so this project has been divided into three different axes, where the first step was to investigate the mechanical compression properties of freeze-drying system, because as you understand here, the idea was not to use classical commercial powder to obtain a tablet, but to use freeze-drying systems. And at this stage of the project, we had very little knowledge about the possibility to use freeze-drying system to obtain tablets. So for the first freeze-drying step, we first had to select our formulation parameters. So for this study, we choose to use and compare two different formulations. The first one containing sucrose as main excipient, and the other one containing trehalose, that both are classically used in the freeze-dried MAP formulation because they present a very good stabilizing agent. Then, so here you can see what is a typical freeze-drying process. So just to remind you that freeze-drying is a dehydration process where the solvent is removed by sublimation, which means that by using this process, there's no heat that could be very detrimental for very fragile biomolecules. And so here you can see the process we choose for this freeze-drying process, which is very common process that are used during the freeze-drying of general molecules, biomolecules more specifically. So then, using these formulation parameters and process matter parameters, we then investigated the physicochemical properties of the obtained freeze-drying systems, and we were able to confirm that by using both of these formulation and process parameters, we were able to obtain freeze-drying system with good properties. First, with a good cake morphology, with a good and expected residual moisture content, and that were both maintained in an amorphous state in the final system. It's important to keep in mind that the obtention of a system in amorphous state is very important for biopharmaceutics because it is the main reasons that can explain why sucrose and trehalose are used as a possess an effective a protective effect when they are used as excipient during freeze drying. So after we confirmed that both of the system with the two formulations were presented good physicochemical properties, the freeze-dried system were used in order to study their tabletability profile, which refers to the ability of powder to be compressed into tablet with adequate mechanical strength. And generally, it is estimated that a tablet with mechanical strength between 1 and 2 megapascal is a cohesive tablet. So here you can see the result, and as you can see, we observed that for both of the formulation, we were able to obtain cohesive tablet even at very low compaction pressure of 25 megapascal, which was a very good news for us because it means that by using very low compaction pressure, we're able to have a good cohesion, which means that we were able to obtain a tablet while applying the lowest mechanical stress on the map for the future. I just here would like to precise that the behavior of freeze-drying system during tableting has been more extensively studied in our labs, and we observed that the freeze-drying system exhibit a very distinct compression behavior comparing to other classical excipients, where it's sometimes necessary to apply very strong compaction pressure to obtain cohesive tablets. Then we further studied the effect of this tablet on the solid state of the systems, and we confirmed that even after tableting, the system still in an amorphous state. And then using them, we also were able to see that the general morphology of the initial freeze-dry powder was conserved even after the tableting. So next, the second step was to investigate now the impact of this tableting process on the biological integrity of MABs, and for that we used an antibody as model for this study that was an antitubulin MAB. So first we studied the effect of this process on the binding properties of the MAB. And so using the indirect ELISA technique, we were able to confirm that for both of the formulation, the binding activity of the MAB, which is here represented as the percentage of enzymatic activity, was not significantly altered after tableting compared to the initial solution control and compared to the condition where the MAB were only freeze-dried. And so demonstrating that the tableting was not detrimental for the binding properties of the MAB. And to go further, we also did some SPR experiments which make possible to precisely quantify the binding affinity of the MAB before and after the process. And for this experiment, we choose to quantify both the binding affinity of the Fab fragments of the MAB, which is the fragments that bind to the target, but also to the other type of the MAB, which is the FC fragment, which is also very important because it is the fragment that is recognized by other immune cells. And as you can see in this table, we were able to confirm that the binding affinity of both types of the fragment of the MAB were fully preserved even after the tableting process. Then we also studied the effect of the tableting on MABs aggregation, which is a big risk for during MAB formulation. And as you can see here, we were able to confirm that after the freeze-drying process, the content of the MAB in its monomeric form, which means that it's when the MAB it's not aggregate, was similar after freeze-drying, and we only observe a minor loss of this monomeric content after tableting that was not supposed to dramatically affect the biological activity of the MAB. Then finally, we also conduct a stability study in order to study the effect of tableting on MAB stability, and we confirmed that the MAB, when formulated into tablet form, was stable for at least 6 months when stored at room temperature. So finally, the last step was to study if this development was applicable when for the formulation of a mucoadhesive tablet. So for that, we choose to add a new excipient in the final formulation that was HPMC. I'm sure you know what is HPMC, but it's a very commonly used excipient that present very excellent mucoadhesive and controlled release properties. And the advantage with HPMC is that it is available in different molecular weight that make us able to control and to choose the mucoadhesive properties we want and the kinetic of the release of the drugs. So first, we developed different formulation of our initial formulation by adding a different concentration of HPMC and with different great, and we study the tabletability profile of this new formulation. And what we observed is that the addition of the HPMC into the initial formulation does not affect the tabletability profile, which means that we're still able to obtain very cohesive tablets even at very low compaction pressure at 25 megapascal. We also studied if the addition of HPMC in the formulation could affect the binding properties of the mAb, but as you can see, we confirmed that the addition of this excipient was not, does not affect the binding properties of the mAb. Then we also conduct a mucoadhesive test in order to determine the mucoadhesive properties of this new formulation. And by using a homemade system, using a mucin tablet that was used to mimic the mucosal surface, we were able to show that depending on the formulation tested, we were able to conserve mucoadhesive properties for at least 24 hours of hydration. And finally, we also made some experiments in order to study the release kinetics of this new formulation. And here also we test different formulation with different percentage of HPMC. And as you can see, depending on the formulation, we were able to see different release times of release, ranging from a few hours to several days. So with this study, we were able to confirm that the development of a mucoadhesive tablet containing a mAb was possible, while maintaining the integrity of the mAb. And just to finish, I would like to present you a project which is currently in development in our lab, and that show how the development of mAbs in tablet form can be particularly interesting. So this is a part of my PhD project, and during my PhD, the aim of this project was to assess the effectiveness of passive immunization as an alternative treatment for the recurrent vulvovaginal candidiasis. So just briefly, vulvovaginal candidiasis is a genital fungal infection that is mainly caused by a species which is Candida albicans. And this is a really common infection because it is estimated that 75% of women will be affected by this infection at least once in their lifetime. And from this woman, it is estimated that 8% suffers from a chronic form, which is called recurrent vulvovaginal candidiasis, and which is characterized by the apparition of at least 4 episodes per year. And the problem is that to date there is no treatments that are efficient to prevent this recurrence. And we know for a long time that this infection is induced by the expression of some fungal virulence factors, but recently we discovered that the infection can also be caused by a local immune dysfunction, while the immune cells that are recruited into the vagina are unable to effectively recognize and eliminate the fungal cells. And that's why in this project we developed 2 different mAbs, where the first one was expected to stimulate the phagocytic activity of the immune cells, and the other one was supposed to modulate and reduce the intensity of the inflammation. And thanks to in vivo study, we demonstrate that the preventive treatment through the intravaginal administration of both of the mAb was effective to protect against the infection by reducing the fungal charge and also the connection in the vagina. So this is a clear example where the development of these maps into tablet form could be very interesting to deliver the map directly into the vagina. So, and just to give you the result we have for the moment, we were able to confirm at this stage that when tableted inside, the co-formulation of the map into tablet form was not problematic, and we were able to maintain the binding properties of both of the maps when tableted. And also we used some different experiments that aim us to confirm that the biological activity of both maps were totally preserved after their development into tablets. So thank you for your attention.
Thank you, Julie. So I don't know if there is some questions for Julie. Perhaps I have one. So you mentioned, so your results is about so antibodies. Do you have also experience on other molecules, other types of biologics being put into tablets?
Myself, no. But in the lab, there are different projects that are in development, and notably by tested the possibility to tablet other biologics such as siRNA and other types of proteins.
Other questions? No? Okay, thank you. So now we will